This is what we've all been waiting for, the last event in the series. I certainly find if I go to 2 seminars in a day, I find it kind of hard to get any work done, even if I find the seminars worthwhile. But we'll plunge on ahead. The original title of my book was Darwinian Agriculture, Where Does Nature's Wisdom Lie? And the editors insisted that I couldn't use a pun in the title. That is, where does nature's wisdom lie? Where is nature's wisdom to be found? But also, where does nature's apparent wisdom lie to us or mislead us? And I argued in the first seminar that natural selection has done a very good job improving trees and not such a good job improving forests, suggesting that if we're looking for useful traits, that natural selection may often have done a good job producing water, flooding tolerant plants with parenchyma and so on, but not necessarily optimizing the traits of a whole ecosystem, such as the level of plant diversity in a lake. And so I'll be talking a bit in this final lecture about some of those ecosystem level issues, things like diversity. But the title of this last talk, Nature Cannot Be Fooled, actually comes from physicist Richard Feynman. It's the last line in his report on the disaster destruction of the Space Shuttle Challenger. And what he did was to go around and talk to NASA engineers and ask them, what's the chance of your system failing and killing everybody aboard? And they would do some calculations and say 1 in 100. And then he would talk to NASA administrators, and they would do a different kind of calculation and come up with 1 in 100,000. So that's where the statement came from: reality must take precedence over public relations because nature cannot be fooled. Now, I don't think we probably have that kind of problem at Erie of lack of communication between the staff and administrators, but I've certainly had the experience of telling some reporter that I made some little scientific discovery, and then there's this headline saying cure for cancer is now imminent. So I think we have to watch out for that, and that will actually occupy a significant fraction of the talk today. So here's an example. I looked at the Erie website before coming here and saw this statement, and this is presumably something that came from Erie scientists, but went through the public relations office, and it makes it sound to me as if C4 rice is probably going to happen within the next 3 or 4 months. Whereas, well, 10 out of 13, right? Whereas in fact, it seems like a project that's well thought out, being conducted well, on track, has a reasonable chance of success. But that this may be perhaps a true statement, but could give a misleading impression of how much progress, how much work remains to be done. Now, I can imagine somebody at a funding agency looking at this and going, well, okay, so will $5,000 be enough to finish it then? So this is one of the themes of my talk today, that we want to avoid misleading statements or unfulfilled promises, exaggerating our accomplishments, or letting the press exaggerate our accomplishments. And these are some of the guidelines I suggest, that if we pay attention to evolutionary trade-offs that will avoid certain kinds of errors, that we want to insist on independent tests. When some corporation says, oh, we've got this wonderful drought-tolerant stuff, it's like, okay, has it been tested by anybody other than you? Often, short-term experiments can give misleading results and make it look like, some genotype or some agronomic practice is better than it, in fact it really is, and longer term experiments will reveal that. I mentioned earlier that looking at overall nutrient budgets is a very useful tool. And finally, I think probably all of you that work in the field have some familiarity with the concept of edge effects, but I think their importance is sometimes underestimated. So, tying this into the three core principles of my book, first, that past natural selection is unlikely to list simple trade-off-free improvements. Certainly, this is a hypothesis subject to disproof by even a single clear counter example, but I think it's at least worth considering when we think about some of the kinds of claims that have been made, particularly for molecular biology. You know, going back to my old buddy Ray Valentine in 1975, suggesting that cereals with nitrogen fixation, might be something that could be achieved in a few years. We're getting on to 40 years now, and maybe we'll do it someday. A number of people have figured out how to make things that look kind of like nodules on the roots of rice or wheat or other crops, and sometimes the press will blow that up as, oh, it looks like we'll have nitrogen-fixing rice any day now. And again, I think it's a very, if it's a step in the right direction at all, it's a very tiny step. The holy grail of increasing photosynthetic efficiency by decreasing photorespiration, Zelich claimed very long time ago to have done this and to have materials with 40% higher photosynthesis. As far as I know, those materials have not led to anything useful in terms of higher crop yield. 40% seems like a lot. Claim, I think with some association with Urey, that transferred one of the C4 enzymes, or maybe two, into rice led to materials with 30 to 35% higher yield. And as far as I'm aware, correct me if I'm wrong, this isn't a claim that's been confirmed through independent testing. And then this high-profile paper in PNAS, where Monsanto announced that they had this wonderful drought tolerance gene. They do have materials that are supposedly drought tolerant that are out being tested, though not independently, but they're not using this gene. As far as I can tell, this one has disappeared from the radar screen again. Correct me if I'm wrong. Well, that's kind of a boring slide. So, one of the things that I've suggested is often useful is to look at things over a longer period of time. And I was sort of up in various places, but that's what I did at UC Davis for most of my time there, was run this long-term experiment. These are half-hectare plots of the bars, little thing there, and my personal interest there was, so I'm interested in nitrogen fixation, is to what extent can we meet the nitrogen needs of non-legumes by growing them in rotation with legume crops that are grown as a green manure. So a mixture of peas and vetch is what it looks like in February. Of course, in Minnesota, it'll be covered with snow, but that's California. And pretty good biomass by the time the crop is mowed and incorporated as a nitrogen source for the future wheat. And if you look at the data for the first few years, it looks pretty encouraging. Here we're comparing the yield over years. The green is the wheat following the green manure legume, and the blue is wheat with an adequate level of fertilizer for rain-fed wheat. And you see in many cases, even a few years in, similar yields between the two. The cases where the wheat after the legume does poorly are cases where legume used up enough water that the wheat was water limited and it's a non-irrigated system. So there's trade-offs there, but just in terms of the nitrogen, it looks like it's meeting all the needs of the wheat. But that conclusion would be premature. I say it's premature. I've seen similar experiments that were a one-year experiment where they concluded, because the corn following the cover crop had good yield, that the cover crop, the legume cover crop or green manure must be meeting all of its needs. But at least we went a few years. But we did include an unfertilized control. And if you look at the unfertilized control, you can see even here, several years into the experiment, that the soil nitrogen, without any added fertilizer, is meeting most of the nitrogen needs of the wheat crop. Now this is California soils, 1% organic matter, but that's still with alternate year harvest. It's a wheat fallow system. That's still, what is it, 500 kilograms of nitrogen per hectare or something like that. It's a good amount of nitrogen that takes quite a while to use up. And so we really can't draw any conclusions from the similar yield of the fertilized wheat and the wheat following the green manure until the yield of the unfertilized control gets much lower, until that's really drawn down, as has happened at Rockstead, for example, after, I don't know, it took 50 years or what, a while. So I mentioned this previously, but this is another useful thing to think about the total nitrogen budget. And I mentioned this particularly in the context of reports from Erie that there's been I would say, an important scientific breakthrough in identifying genes that lead to greater phosphorus uptake, apparently due to a more larger, more robust root system. But phosphorus taken up this year is not going to be available next year. So the question is this really going to be, how useful is this going to be in low phosphorus input systems? It seems like you still need to place the phosphorus that's harvested. But there are a number of people that have made the same kind of suggestion. We've got some way of increasing phosphorus uptake, and that will solve the problem that phosphorus ores are running out and so on. Citrate exudation, something that plants, a variety of plants do to some extent. Plants with proteoid roots actually have a supercharged version of this, and they can solubilize and take up phosphorus that wouldn't otherwise be available. But the question is, how long is that pool of phosphorus going to last before it's all been mined out? So you really would need to look at a multi-year experiment. It might do great in 1 year, but how's it going to do over a longer period of time? Same argument for mycorrhizae. They can take up more phosphorus, but they don't create phosphorus in the way that rhizobia create nitrogen, for example. Okay, well, the slide where I showed the yield data from the Erie rice phosphorus stuff has disappeared along with one of my other slides. I don't know what the problem is here. But the bottom line on that is a couple of things. First, the particular gene enhanced phosphorus uptake in IR72. In IR64, the transgenic plant with the gene had higher yield in a low phosphorus environment, but so did the null control that had, I don't know what, just the cassette without the actual gene, so it didn't seem to work there. And the yield data, which were presented there, were actually measurements of 3 plants per replicant. And so when I talk about edge effects, I think you need to, there's a matter of sort of possible inadvertent sampling bias and all that sort of thing. So when I think about yield, I really like to see yield of a large plot and not the rate of a few plants. But let me turn to my second point, which I haven't talked about that much, that nothing has consistently improved the organization of natural ecosystems. I devote a whole chapter to trying to make that point. And so if that's true, you know, we can look at natural ecosystems like this tropical rainforest in Australia has very high species diversity. And if natural selection or some other, you know, self-organizing or succession or some natural process adjusted the number of species in an ecosystem up or down in order to get just the right level of diversity to maximize productivity, resource use efficiency, stability from year to year, long-term sustainability and resource use efficiency and so on. If there were some mechanism that did that, then if we were farming near this tropical rainforest, we'd say, well, we ought to have the same number of species in our orchard that they have in the tropical rainforest because we're copying a useful, reliable model. But if, as I argue, there is no such process, then it's still useful, very useful to study these natural ecosystems and look at how diversity, for example, affects all of those things I talked about. But we can't just assume that the level of diversity we see in a natural ecosystem is optimal. And so that's useful because often in natural ecosystems we have mixtures of species living together, although that wasn't true for the wild rice case, and apparently it's not true for the wild ancestors of wheat living in Uzbekistan or wherever they are. But many natural ecosystems have relatively high diversity, so if we were to copy them, we might want to do something like this and have an intercrop of corn and beans, for example. But I would argue that we need to also consider ways of using diversity that are different than the ways we see in nature. You don't see, this is, you know, illustrating crop rotation, you know, this year we have potatoes here, but next year it's going to be barley, and the year after that will be peas or whatever it is. And that's something you don't see in the patterns of natural ecosystems. You don't have maples this year, and the next year they're all oaks, and then the year after that they're all pines. But if nature hasn't perfected the organization of natural ecosystems, that isn't an argument against crop rotation. And this is just basically reiterating my logic that natural selection has given us useful traits in individual plants and animals because these plants have had to compete against each other, and the ones that had the right size of a parenchyma won in the competition and passed that trait on to their descendants. But lakes with low diversity, like this natural lake in Minnesota, haven't had to compete against lakes with high diversity, and even if they did, they don't have any way to pass their diversity in some sort of exact copy like we get with DNA-based heredity onto their descendants. My third point can be expressed two ways. Either we need more diversity and it has benefits, but it also has a cost, but we're willing to pay that cost, or there's a cost, but it also has benefits, but we're willing to, we should often pay that cost. And so basically the cost is that if you use diversity to minimize the risk of total failure, you're also giving up the opportunity to have the maximum possible, because your maximum possible productivity would be to grow the single most productive genotype. But that would entail risks that would be reduced by having a mixture of genotypes, but likely that mixture is going to have lower, when I say mixture, I don't necessarily mean mixed together in a field, but growing a variety of things, some of them will have lower yield than the maximum. So when we talk about diversity, it is sometimes used by my hippie friends as a code word for intercropping. And so I'm saying that we need to increase diversity, but we need to actually think long and hard about how we're going to deploy that diversity in space and time. So at the global and regional scale, I think having a variety of crop species and a variety of genotypes within those crops is critical to minimizing the risk of disastrous food shortages. And because Erie has all these connections, I think you could probably play a significant role, and in fact are playing a significant role in this. So this is probably not news to anybody here in the room, but for those of you out in YouTube land, the global food supply rests really heavily on these three crops: wheat, rice, and maize. And these are all great crops. But it seems risky to me to have so much reliance on so few crops, even though there's a lot of diversity within each of these. I wonder if we might not be better off if we had a few more potatoes and more grazing-based dairy system and sweet potatoes and cassava and things that aren't even on here, like buckwheat and so on, having a larger role. So I don't know whether the International Rice Research Institute wants to. You're broadening your perspective a bit with this ecological intensification system with maize and possibly soybean. I think that's probably a good idea, but maybe you should be the International Rice Growing Regions Crop Production Institute or something like that. And you don't want to neglect rice, of course, but perhaps think about greater crop diversity, how you contribute to that. But as I said earlier, advocates of diversity sometimes really are talking about the kind of intermittent mixtures of plant species or perhaps of genotypes that we see in nature. And I would argue that the benefits of those mixtures in reducing disease and in various kinds of yield benefits from complementarity, that those benefits are real, but that they may have been exaggerated relative to crop rotation, which is another way of losing diversity. And so let me illustrate that with this cartoon. So on the right, I have an intercrop of, I don't know, rice and wheat, perhaps. And on the left, I have whole fields of rice and wheat. And so the question is, which of these is going to be better in terms of limiting the spread of fungal disease, for example? Well, if you've got some, rice disease, rice pathogen here in this rice field, it's going to spread, if you've got only a single genotype, it's going to spread from plant to plant to plant to plant to plant very rapidly through this field. But then, hits the wheat field, and there's a big barrier before it can get to another rice field. It has to sort of sneak through at the corners, and maybe the wind isn't blowing that direction. Now here, you're going to have slower spread because fungal spore from a rice plant, a fair number of them are going to land on the wheat plant, and they're going to die there and not spread as rapidly. So there's definitely this benefit to reducing the spread of disease, but to the extent that they can spread, there's no barrier. They just can keep on going. So that's talking about the spread within a year. But then if we think about pathogens that overwinter in the soil, then we have to think about crop rotation. And if you look at the upper left, you see here it is in rice this year, next year it's in wheat, next year it's in rice, next year it's in wheat. And so if you have pathogens that tend to die out in the soil, they will die out between successive rice crops, whereas over here in the intercrop, you've got rice every year, so you won't get that benefit of rotation. Now you may argue, well, we'll use 4 species of 2 species, and then we'll rotate that with another intercrop of 2 different species. Well, that's fine, but you could take those 4 species and have a 4-year rotation. Now, which of those would be better? And I'm not actually going to answer that question. I'm just saying that we should be thinking about that. We shouldn't be thinking about diversity as necessarily involving intimate mixtures of species. Think about the big picture here. So, what I would really like to see, and if anybody either here at Erie or out in YouTube land knows of such a study, I'd be really interested in a case where somebody has actually compared an intercrop of 2 species with a rotation of those same 2 species. And this would have to be a multi-year experiment. If it's a 2-year rotation, you've got to go at least 4 years before you can draw any conclusions. And that's, a pretty long, master's degree project. If it's a 3-year or 3 species intercrop, then you're talking about a 3-year rotation or 6 years to test it out. So I think that's something that needs to be done, but I've never seen it. I've seen many, many 1-year comparisons of intercropping with monoculture, but not comparisons between crop rotation and intercrops of the same species. So in the book, I rely on a computer model from a Dr. Zhu, who kindly sent me the data, and he essentially modeled what we have here, plus with the one on the left being the crop rotation, the one on the right being the intercrop, and they also modeled a monoculture where the whole field was 1 species year after year, or 1 genotype year after year. And then he modeled the level of disease, so higher levels are bad over a number of successive seasons with carryover in the cases where you didn't have crop rotation. And in the first year, you get what you always see in intercropping experiments. The rotation treatment, was these solid blocks of 1 species, and so they had faster spread of the pathogen, and the lowest levels of the pathogen were in the intercrop or the high diversity plots. But when we go to a second year and have carryover of pathogens, that pattern is reduced, and the lowest level of disease is in the rotation. Then you have a year where they're similar, another year where they're similar, and then another year where the rotation does a little bit better. Now, this obviously was a stochastic model. That's why you're seeing these kind of strange changes from year to year. But I think this modeling approach is certainly very useful and worthwhile, and can be used to explore other options for deploying diversity. But I would really like to see experimental data